Good morning to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, in today's session are 100 concepts for NEET 2026, wherein we have dealt with the concepts of light reactions, early discoveries, cyclic and non-cyclic photophosphorylation, chemiosmotic hypothesis. Then we have started with light independent reactions, where we have completed C3 cycle in yesterday's class, and we have also covered up the photorespiration. In today's class, we are going to completely in detail study about C4 cycle, CAM cycle and C4's Kranz anatomy specially, and we are going to draw out the clear differences between C3, C4 and CAM cycles, okay, which is given in our NCERT. Those three we are going to draw out today. So first, if you have to start C4 cycle, okay, C4 cycle, which is discovered by Marshall Davidson Hatch and Charles Loger Slack. That is why this C4 cycle is also called as Hatch and Slack pathway. Okay, this is also called as Hatch and Slack pathway. Fine, because of their discoveries from Marshall Davidson Hatch and Charles Roger Slack in the year 1966. So this is a modified carbon dioxide fixation pathway. We are going to learn it, and here the first stable product formed is 4-carbon oxaloacetic acid. As the first product that is formed is 4-carbon oxaloacetic acid. The name is given as what C4 cycle. Okay, so here the C4 cycle, in the C4 pathway, both C3 and C4 cycles are involved, wherein C4 cycle is conducted in the mesophyll cells. Okay. Here, before learning about both C3 and C4 and what is the mechanism of action of this C4 cycle, we have to be clear about the Kranz anatomy. So, what is Kranz anatomy? Fine. Kranz anatomy is only seen in C4 plants. C4 plants are what plants? Tropical plants. Okay, in tropical plants, those are C4 plants, wherein Kranz anatomy is observed. Best examples for this can be what? Sorghum, maize and sugarcane. These are the best examples, okay, in where C4 cycle will be conducted. The only dicot in which C4 cycle is conducted is what? Amaranthus. Okay, in Amaranthus, C4 cycle is conducted. Okay, this is the only dicot. Mostly in monocots, C4 cycle will be conducted. Now let us look into the Kranz anatomy. Usually in C3 plants or C4 plants, you see vascular bundles are covered by what? Bundle sheath cells. That is clear to us, right? Vascular bundles which are conjoined and closed, right? Conjoined and closed vascular bundles in the leaf. They are surrounded by what? The bundle sheath cells, both in C4 plants and C3 plants. But then in C4 plants, the vascular tissue which is surrounded by the bundle sheath cells, this is of many layers and there is no intercellular space in the bundle sheath cells. Okay, many layers of bundle sheath cells will be surrounded around the vascular bundles. These bundle sheath cells will be having agranule chloroplast beta. Okay, these bundle sheath cells will be having agranule chloroplast. Let me give you in detail. Okay, so these bundle sheath cells of C4 plants will show you what? Kranz anatomy, of course. Kranz anatomy, and they have agranule chloroplast. They have agranule chloroplast. Agranule chloroplast. Agranule chloroplast, which means it has stroma lamellae. Okay, it has what? Stroma lamellae. With this stroma lamellae, it will be conducting what? Cyclic photophosphorylation, where there is no involvement of photolysis of water. Okay. So, with this stroma lamellae, it will be conducting what beta? Cyclic photophosphorylation. Photophosphorylation. Okay, cyclic photophosphorylation, and there is no photolysis of water. Photolysis of water. And as these bundle sheath cells are deeper inside the mesophyll cells, okay, in their stroma they have what? Rubisco. Okay, stroma will be having rubisco. Stroma which is having rubisco is conducting C3 cycle or Kelvin cycle inside the bundle sheath cells. Fine ma. The bundle sheath cells are having agranal chloroplast where there is no grana are present, which is having agranal chloroplast which does not have grana, whereas this is having stroma lamellae which is conducting what? Cyclic photophosphorylation, because it has stroma lamellae and it has what? PSMA? Photosystem 1 only, where the maximum wavelength is what? P700, and it conducts what? Cyclic photophosphorylation, and it does not have photolysis of water. Photolysis of water does not take place over here. And inside the stroma, if you see, inside the stroma of the bundle sheath cells chloroplast, you can observe rubisco. This is conducting C3 cycle or Kelvin cycle. So, here if you see, C4 plants will exhibit several characteristics that help to prevent what? Oxygenation of rubisco. In C3 plants or in C3 cycle of C3 plants, we have looked into the rubisco which is showing dual nature, both oxygenase activity and carboxylase activity. When oxygenase is activated, that is conducting photorespiration. But here, because of this bundle sheath cells having rubisco and conducting C3 cycle, this is preventing the photorespiration. Okay, this is prevents photorespiration. Fine ma. So, here if you see, in C4 plants photorespiration does not occur, because in the mesophyll cells peptidase enzyme will be there which will only bind to what? Which will only has carboxylase activity. Deeper inside the bundle sheath cells rubisco will be present, whereas it is of course it can bind to carboxylase oxygenase, but deeper inside the cells of bundle sheath cells only carbon dioxide will be available. So, there is no possibility of oxygenase activity over here. So, this is an adaptation which is taken by C4 plants, tropical plants, to escape the process of photorespiration. So, strategies to prevent the oxygenation of rubisco. Look into the theory properly ma. Whatever we have discussed is over here. Agranal chloroplast, they are found in bundle sheath cells of C4 plants which prefers cyclic photophosphorylation, as I said you, and this prevents the photolysis and photorespiration, both are prevented. Okay, no photolysis because there is no grana. And here if you see interior location of bundle sheath cells in a plant, this keeps them at a low, relatively lower temperature. This keeps them at low temperature, and bundle sheath cells will have thicker walls. Bundle sheath cells will have thicker walls, thicker cell walls, and there are no intercellular spaces, which this will prevent the gaseous exchange of oxygen. This will prefer for what? Gaseous exchange of oxygen, and this will protect what? Rubisco, which is present inside the bundle sheath cells. Next, Kranz anatomy. Kranz anatomy, this is a setup in which bundle sheath cells will form several layers around the vascular bundles, and this characterized by thick walls and there is no intercellular spaces, and there are large number of chloroplasts inside the bundle sheath cells which are agranal chloroplasts. The thick walls, no intercellular spaces are present in between them, between the bundle sheath cells. This will prevent the entry of oxygen and this will escape the carbon dioxide. So, this is the event that is happening inside the bundle sheath cells. Till here it is clear, Noma? Now, Kranz anatomy is clear to us. The strategies what C4 plants are following to escape the oxygenase activity of Rubisco are clear to us. It is very clear to us about the bundle sheath cells which are of several layers around the vascular bundles in C4 plants, which is preventing the oxygenase activity of Rubisco, and this further conducts what? C3 cycle. Okay, and the stroma lamellae which is present in the chloroplast of bundle sheath cells, that stroma lamellae is preventing photolysis of water, conducting cyclic photophosphorylation, and photorespiration is also prevented in bundle sheath cells. So, here if you see in C4 cycle, okay, in C4 cycle if you have to see, first is carbon fixation. Okay, C4 cycle first step is what? Carboxylation or carbon fixation. This is carboxylation or carbon fixation. Carboxylation which is happening in the mesophyll cells. What happens here? First carbon dioxide will enter into the mesophyll cell. We know, right? So, if you consider this as a leaf, so how carbon dioxide enters into the leaf? Through stomata. Through stomata, carbon dioxide enters into the mesophyll cell inside the chloroplast stroma. Carbon dioxide will be turned to hydrogen carbonate by the enzyme carbonic anhydrase. Okay, it gets turned to hydrogen carbonate by the enzyme carbonic anhydrase. So, this hydrogen carbonate along with PEP by the enzyme pepcase. So, here what is the primary acceptor of carbon dioxide in C4 cycle? Primary acceptor will be hydrogen, primary acceptor will be PEPMA. Phosphoenol pyruvate is the primary acceptor of carbon dioxide in C4 cycle. Pepcase, phosphoenol pyruvate carboxylase. So, pepcase is the first enzyme to catalyze the carboxylation or carbon fixation, like how we have Rubisco in C3 plants. Here in C4 we have pepcase. As such, the primary product, yes, Saurabh Gupta, yes, you can ask what is the question? You can ask the question, Vita. So, oxaloacetic acid will be turned to what? Malic acid or aspartic acid. So, here the primary product of C4 cycle is what? Oxaloacetic acid. Okay, and this getting converted to malic acid or aspartic acid is catalyzed by the enzyme malate dehydrogenase. Okay, so this is catalyzed by the enzyme malate dehydrogenase. Either malic acid or aspartic acid which is formed, this will enter into the bundle sheath cells through plasmodesmatal connections. Okay, how they are linked They are linked by plasmodesmatal connections. Mesophyll cell and bundle sheath cells are connected by plasmodesmatal connections. So, here with the help of plasmodesmatal connections, either malic acid or aspartic acid will be entering into the bundle sheath cells. Primary what happens in the bundle sheath cells is decarboxylation. This 4-carbon compound will be decarboxylated to 3-carbon pyruvate and 1 carbon dioxide. This 3-carbon pyruvate will be regenerated to PEP, wherein from bundle sheath cells that it is regenerated to phosphoenol pyruvate. Okay. For this regeneration step, it is utilizing 2 ATP. Okay. It is utilizing how much ATP 2 ATP, whereas that 1 carbon dioxide which is present in the bundle sheath cells, this will be conducting what cycle? Kelvin cycle. And in Kelvin cycle we know in the reduction step of Kelvin cycle 2 ATP are used and in the regeneration 1 ATP is used. As such, for 1 carbon dioxide totally 3 ATP are used and 2 NADPH in reduction process. On total for C4 cycle 5 ATP, 3 in the Kelvin cycle and 2 in the regeneration step, on total 5 ATP and 2 NADPH will be present, will be utilized for the C4 cycle. So, here once again let me give you a gist of C4 cycle. In the mesophyll cells, when carbon dioxide is accepted, it first converts to what? Hydrogen carbonate by the enzyme carbonic anhydrase. This is accepted by the primary acceptor, that is phosphoenol pyruvate, and this is catalyzed by the catalyzer, that is phosphoenol pyruvate carboxylase, and the primary product formed is what ma? Oxaloacetic acid. This turns to malic acid or aspartic acid by the enzyme malate dehydrogenase. They will be delivered to what? Bundle sheath cells by plasmodesmatal connections, wherein this 4 carbon compound is decarboxylated to 3 carbon and 1 carbon. 3 carbon pyruvate is regenerated back by utilizing 2 ATP into PEP, and this 1 carbon dioxide will run the cycle, Kelvin cycle, wherein 3 ATP and 2 NADPH will be utilized. So, on total if we have to calculate beta, on total for 1 carbon dioxide fixation in C4 cycle, let me draw out the energetics for you. So, here let us look into bioenergetics of C4 cycle. Fine ma. So, let us look into that. First, for 1 carbon dioxide we will see, okay. For 1 carbon dioxide fixation, how much ATP is utilized beta? First 3 ATP and 2 NADPH. Where are they utilized? In C3 cycle, and 2 ATP for the process of regeneration. Clear, right ma? So, here as such, to form 1 glucose molecule, how much ATP will be utilized ma? For 1 glucose, on total it is 5 ATP, right? 5 ATP for 1 carbon dioxide fixation. To form 1 glucose, we all know how many cycles have to turn? 6 cycles. So, 6 carbon dioxide fixations have to happen. For 1 carbon dioxide it is 5 ATP. So, for 6 carbon dioxide it is 6 into 5. How much beta? 30 ATP. On total how much? 30 ATP will be utilized, and NADPH will be common, that is 12 NADPH for the formation of 1 glucose. Now, here comes the question, okay, how much amount of extra ATP are consumed by C4 cycle compared to C3 cycle? This is one of the application question. For example, for 1 carbon dioxide extra ATP, let us look into the extra ATP concept. So, here we will look into the extra ATP consumed by C4 cycle. Okay, in this extra ATP, if you see ma, for 1 carbon dioxide, extra ATP utilized are how much? 2 ATP by C4 cycle, that is for the process of regeneration. For how much this 6 carbon dioxide fixation, or for 1 glucose? For 1 glucose, how much extra ATP are utilized? If that is the question, it is 2 into 6, that is 12 ATP. 12 ATP are extra, okay, those are utilized by C4 cycle. Clear, right ma? So, this is all about the Kranz anatomy of C4 plants, mechanism or physiology on how C4 cycle is conducting, and this is the bioenergetics of C4 cycle. Clear with it, right? Now, we will be looking, this is all the gist which I have explained you, just go through it. Whichever I have explained is over here ma. So, let me read out to you once, it will be clear for you na. So, phases of C4 cycle. First is what? Initial, that is carbon fixation. Carbon dioxide acceptor is what? 3 carbon compound, that is phosphoenol pyruvate. Second, the enzyme responsible for this carbon dioxide fixation is what? Pepcase or phosphoenol pyruvate carboxylase. Third question, mesophyll cells lack the enzyme rubisco, true. Oxaloacetic acid is formed in the cytosol, correct? Oxaloacetic acid then gets converted to what? Malic acid or aspartic acid. This occurs in mesophyll itself. Transport, that is decarboxylation and then transport, this happens where? In the bundle sheath cells. Carbon dioxide acceptor is 3 carbon molecule, that is PEP. Malic acid or aspartic acid are translocated to where? Bundle sheath cells. Carbon dioxide is removed from malic acid or aspartic acid to enter to Kelvin cycle. Next, regeneration of phosphoenol pyruvate. 3 carbon compound is transported back to mesophyll cells where it is converted to PEP again. This is done with the help of sensitive enzyme, cold sensitive enzyme, that is PEP synthase. Do remember those enzymes which are involved in dark reaction beta, like it can be pepcase or phosphoenol pyruvate synthase, or it can be PEP carboxylase or rubisco. These are temperature sensitive enzymes. When we are looking into the factors affecting photosynthesis, we will be dealing with that more clearly. So, here this is done with the help of what? Cold sensitive enzyme, that is pepcase. Clear? This is done with cold sensitive enzyme, that is pepcase. Now, let us look into the CAM cycle. In C4 cycle, okay, 2 cells are involved, mesophyll cells and bundle sheath cells, right ma? And in CAM cycle here, the only difference is both C3 cycle and C4 cycle will be happening in the mesophyll cells only, but the only difference is primary carboxylation or the first carbon fixation, initial carbon fixation takes place during the night time and second carboxylation will take place during daytime. So, here you see the CAM cycle, crustacean acid metabolism, wherein this is a photosynthetic adaptation in succulent plants, okay, for periodic water supply, wherein plants in arid regions, tropical epiphytes will be having scotoactive stomata. What is this scotoactive stomata beta? Scotoactive stomata means stomata opens during night time. Stomata opens during night and close during day. Night and close during daytime. Okay, this is called as what beta? Scotoactive stomata. Scotoactive stomata. Clear, right ma? So, here we have scotoactive stomata where stomata opens during night and closes during daytime. So, first carboxylation occurs at night time. One question ma'am. Yes, Saurabh Gupta, you can ask me the question. Just text the question, I will be answering you. Carbon dioxide by the help of carbonic anhydrase, when stomata is open during the night time, carbon dioxide will enter and by the help of carbonic anhydrates, it gets turned to hydrogen carbonate accepted by the primary acceptor, that is PEP. By the help of pepcase, it is fixed to OA, which is formed to malic acid or aspartic acid by the enzyme malate dehydrogenase or aspartic dehydrogenase. And this malic acid will be entered into the vacuole, and in vacuole it is stored all during night. During daytime, this malic acid will be decarboxylated to pyruvate and carbon dioxide. This carbon dioxide will run C3 cycle. Okay, carbon dioxide will be run C3 cycle, whereas pyruvate is regenerated back to PEP by the enzyme PEP synthase, and it is utilizing how much? 2 ATP. Okay, it will be utilizing how much? 2 ATP. So, look at here, okay, scotoactive stomata, CAM plants will be having scotoactive stomata. Mostly these are the desert plants or plants belonging to arid regions and tropical epiphytes. So, they will be conducting, they will be having scotoactive stomata and initial carboxylation during night time, and second carbon fixation, that is C3 cycle, will happen during daytime. Bioenergetics are same as that of C4 plants, only difference there 2 cells are involved, mesophyll cells and bundle sheath cells. Here only one cell, that is mesophyll cells. There carboxylation takes place during daytime only, but here first initial carboxylation during night time, second during daytime. Okay. So, this is because of scotoactive stomata. This is all about C4 cycle and CAM cycle. Now, at the end, let us look into this, which is very important, this table which is given in our NCERT. Let us choose the comparative analysis for all of this, wherein we can expect a questionnaire from this table. Okay. First characteristic, that is cell type in which Kelvin cycle takes place. Kelvin cycle in C3 plants takes place where beta? In mesophyll cells. In C4 plants it will take place in bundle sheath cells. CAM plants also it will be taking place where? In the mesophyll cells only. Okay. Cell type in which the initial carboxylation reaction occurs. Where does initial carboxylation reaction occurs in C3 plants? In mesophyll cells. In C4 plants, where is the initial carboxylation? In C4 plants also it is mesophyll cells, CAM plants also it is mesophyll cells. How many cell types does the leaf have that fix carbon dioxide? Here it has two cell types, mesophyll cells and bundle sheath cells. Here it has only one cell type, that is mesophyll cell. CAM plants are also having only one cell type, that is mesophyll cell. Which is the primary carbon dioxide acceptor? Primary carbon dioxide acceptor in C3 plants is rubisco. Okay. Primary carbon dioxide acceptor in C4 plants is, here he is asking you about the acceptor, not enzyme, Here the primary acceptor in C3 will be RUBP, in C4 it will be PEP. Okay. And here in CAM plants also it is phosphoenol pyruvate only. Okay. Primary carbon fixation. Okay. Number of carbons in the primary carbon dioxide acceptor. Okay. In primary carbon dioxide, number of carbons in RUBP are 5. Here it will be 3 carbons. Here also it is 3 in CAM plants. Here you look into which is the primary carbon dioxide fixation product. The primary carbon dioxide fixation product in C3 plants is 3 carbon compound, that is phosphoglyceric acid, and here in C4 plants that is 4 carbon compound, that is oxaloacetic acid. Here it is phosphoglyceric acid, and here also in CAM plants it is 4 carbon compound. What is it beta? Oxaloacetic acid. Okay. And number of carbons in the primary carbon dioxide fixation product. Okay. How many number of carbons in the first carbon dioxide fixation product it has? This is 3 carbon, this is 4 carbon. Here also in CAM plants we will have 4 carbon. Does the plant have rubisco? In C3 plants it has, C4 has, CAM also has rubisco. So, C3 cycle is seen in all the plants. Okay. C3 cycle is observed in all the plants. Does the plant have pepcase? No, C3 plants does not have, but C4 and CAM plants will have pepcase. Which cells in the plant have rubisco? Which cells is having here? Mesophyll cells. Here bundle sheath cells in C4 plants, again in mesophyll cells of CAM plants. Next, carbon dioxide fixation rate under high light conditions. Under high light conditions, carbon dioxide fixation rate under high light conditions. High light intensity, carbon dioxide will be solubilized, and rubisco which is present over here will does oxygenase activity. Okay. So, it does what? Oxygenase activity. So, here carbon dioxide fixation at high light conditions, here rubisco will be low because it conducts oxygenase activity. In C4 and CAM it will be high only. So, this from these questions like whether photorespiration is present at low light intensities? No, photorespiration is not present at low light intensities. It is present at high light intensities only in the C3 plants, not in the C4 and CAM. And here whether photorespiration would present at low carbon dioxide concentration? Yes, when there is low carbon dioxide concentration there will be high oxygen and rubisco will conduct oxygenase activity. Here it is C4 it is correct, in C3 it is correct, C4 and CAM there is no possibility of photorespiration only. Whether photorespiration would be present at high carbon dioxide concentration? No, photorespiration will not be in high carbon dioxide concentration. Temperature optimum in C3 plants, as they are temperate plants, it will be less than, it is 25 to 30 degrees Celsius. In C4 plants, as they are tropical plants, it can be approximately 40 degrees Celsius to 45. CAM plants, it can even be above 40 degree Celsius. So, here it in C4 it is 30 to 40, 20 to 25 in C3 plants. Okay. C3 are temperate, C4 tropical, CAM plants, desert plants which can grow more than 40 degree Celsius. C3 plants best examples can be tomato, bell pepper, rice, wheat. These are what? C3 plants. Whereas C4 plants which are tropical plants, this can be sorghum, maize, sugarcane, okay, amaranthus. These are the best examples of C4 plants. CAM plants, opuntia, cactus, okay, epiphytes, all this belongs to CAM plants, like those plants which belongs to Crassulaceae family. Okay. So, with this we are done with C3, C4 and CAM. In tomorrow's class we will be looking into the factors affecting photosynthesis. So, before summing up the class, I would like to give you a brief intro about our NEET World's special programs, wherein we have NEET World's 2 steps to medicine, NEET World's 1 step to medicine and NEET World's power step to medicine, wherein the fee is very minimal for all three courses, that is 3333 per month, wherein you can directly call us to the given number to know more about these courses, or you can visit our branch which is in Narayanguda, Hyderabad. And lastly, to all the students who are learning in the public platform or who are preparing on yourself for NEET 2026, we have our NEET World's Telegram group to support you in all the extents by providing you the daily quizzes of biology, chemistry, physics and mathematics, and by updating you about the live class schedules, and by giving you the free resources, finally providing you one-on-one doubts, faculty solving sessions. Lastly, how you can join this Telegram group is just by scanning this QR code or visiting our official website, that is www.neetworld.in. Okay. So, this is all about our Telegram group beta. See you in tomorrow's class. Be prepared, be strong and confident, okay, and continue your preparation happily. Thank you.